I'm delighted to introduce the Provost, Eric Kaler, who is going to introduce tonight's speakers.
Thank you, Jeff, and good evening, distinguished colleagues and guests. It's really a great pleasure for me to be here at this lecture. These lectures sponsored by my office, and this one in partnership with the Department of Ecology and Evolution and the Living World Lecture Series are an important part of the intellectual life of Stony Brook, and I'm very pleased that you're able to be here for this. It's also a thrill to introduce our keynote speaker, Dr. Douglas Futuyma, who is a distinguished professor of ecology and evolution at Stony Brook. Doug received his PhD from the University of Michigan in 1969, and his research interests focus on speciation and the evolution of ecological interactions between species. He was a Guggenheim Fellow and a Fulbright Senior Scholar, and served as president of the Society for the Study of Evolution, the American Society of Naturalists, and the American Institute of Biological Sciences. I presume, Doug, not all at the same time, but I don't know. Okay, good, different years. He was elected to the National Academy of Arts and Sciences in 1996, and to the American Academy of Arts and Sciences in 1996, and the National Academy of Sciences in 2006. He's the author of the textbooks Evolutionary Biology, Evolution, and Science on Trial, The Case for Evolution. He also serves as editor of the Annual Review of Ecology, Evolution, and Systematics. Last year, as you know, marked the 200th birthday of Charles Darwin and the 150th anniversary of On the Origin of Species. Professor Futuyma's talk is certainly well-timed as we move forward from these milestones and celebrate Darwin and those who continue his journey. Accordingly, in honor of the life and work of Charles Darwin, Douglas Futuyma will speak tonight on evolutionary biology, 150 years of progress, and he'll describe some extraordinary achievements since the publication of Darwin's book. I'm very honored to have our distinguished colleague speak tonight, and I'm anxious to hear what he has to say. So please join me in welcoming Professor Futuyma to the podium.
I'm not sure if I'm wired with several microphones, and I'm not sure if they're all on. Thank you very much, Provost Keller, for your kind introduction, and thank you all for coming tonight. I had thought that perhaps after living through 2009, the Darwin year, that everyone might have seen so many programs and read so many articles and perhaps gone to so many other events commemorating Darwin and his impact on Western thought, really, that you might be quite saturated and you might want to hear nothing whatever about Darwin or evolution for at least another 5 years. So thank you very much for coming out. I just noticed that the title that I put as my title slide is not the same as the title on either of two posters that can be found around campus, which themselves have two different titles. One of them, which I believe I supplied both of those titles to different people, to the Provost's office and to Jeff Levinton. So let me just review those titles. One poster, the title is Evolution, 150 Years of Progress. And you think, well, really, has there been very much progress in evolution in 150 years? And that actually turns out to be a very big question, because it's really a philosophical issue as to whether or not you can define any progress whatever in the entire history of evolution. The word progress is a very loaded word, and we could have quite a long conversation about that. The other title, which Provost Keller announced, is, I think, more accurate. I want to speak about evolutionary biology, 150 years of progress. And here we are indeed talking about an immense amount of progress since Darwin published the book On the Origin of Species in 1859. There are quite a few people, quite a few evolutionary biologists who were really rather uncomfortable about all of the Darwin hoopla last year because they felt as if he was almost treating him as a cult figure. You know, we don't have a Newton Day or a year of Lavoisier or even of Einstein, but for some reason Darwin is the name is what? The name is a symbol, right? It's come to mark a whole dimension of thought and to some extent a whole worldview in the eyes of certain people in a way that you would not associate with Lavoisier or even Einstein. And Darwin is both a demonic symbol to a very large fraction of the American public, and then perhaps in response to that, a symbol of the power of science and the power of rationality within the world of people who think that we should think rationally and come to conclusions based on evidence. Darwin, in fact, would not be able to understand most of the lectures that I and my colleagues give in our courses today in any field of biology, including evolutionary biology, because the field has moved on so much in so many different ways. So Darwin may symbolize the field, but the fact is that we have made immense strides in many ways. And I would happily keep you here until 11 or 12 o'clock tonight talking about the many ways in which evolutionary biology has progressed. But I will do my best not to do that. Let's very quickly see what did Darwin give us, okay? And then this is the launching point. And people talk about Darwin's theory of evolution, but Ernst Mayr, one of the leading figures in evolutionary biology in the last century, pointed out that really you can recognize a number of different theories in the origin of species. And with slight modification, these are the points that Mayr makes. First of all, Darwin he had a theory of descent with modification. That is to say, he claimed that all species of organisms have probably evolved from a single original common ancestor, giving rise to many different lineages through time, which have become different, each of which has become modified through the course of time so that they differ immensely from one another. So this is the claim that we are united by bonds of descent with every other organism in the world, that we are really related not just to other mammals, but to fungi, to mushrooms, to bacteria. And this has surely got to be one of the boldest and most outrageous hypotheses ever offered by anyone in the history of thought, in the history of science. Number two, so that is a claim about the history of evolution. Okay, that evolutionary has consisted of a series of branching processes giving rise to different lineages from common ancestors, and each of those lineages undergoing modification in its characteristics over the course of time. So how does that modification take place? And here's when we get into what we can call evolutionary theory in the current use of that word, where we're talking about what are the processes, the causes of evolutionary change. And according to Darwin, the evolutionary modification, whoops, I didn't intend to do that. Evolutionary modification is a two-step process. First, he said, there is variation among individual organisms of a species in characteristics, whether it's height or coloration or the length of the nose or whatever. This inherited variation arises by mechanisms he had no glimmering of. But he says it arises, and he believed that often these new variants arise independently of their adaptive value or of the organism's needs. Okay, so that is, in some sense, a separate hypothesis, isn't it? But then he said these inherited variations arise, and then a rare variant may increase in frequency in the population. What does that mean? It means that first only a few individuals of the species or of the species population carry this particular variation, but that by virtue of their improved survival or their improved capacity to reproduce, they will end up having more descendants per capita than individuals that don't have that advantageous variation. And so over the course of time, the species will be transformed as an initially rare advantageous variant form becomes more and more common and replaces the ancestral form, let's say the ancestral color type. So you might have a replacement of a pale gray type by a dark black type, and it's the percentage of black types increasing over time. So in other words then, and this was critical, that this evolutionary change is a modification of the population. Okay? The first appearance of this variation, the first black individual in a population of gray moths, for example, is not a new species. It is not really, the species hasn't evolved yet until that new form, which happens to be advantageous, let's say in an industrially polluted environment, is able to survive or reproduce better. And so the cause of this gradual replacement of one form by another within a species is, the cause of that is a consistent difference in their rate of reproduction or survival, and that is what he called natural selection. And finally, Darwin postulated that in general, new variations, if they dramatically and drastically alter the organism, are usually disadvantageous. He thought that advantageous what we would today call advantageous mutations, advantageous variations, are those that would not disrupt the whole organism or the organization of a plant or animal, but rather would be a relatively minor change in some characteristic. And therefore, Darwin postulated that evolutionary changes over the long time may be quite great, but that they result from the sequential accumulation of slight differences, little bit by bit by bit, over the course of vast periods of evolutionary time. So these were basically Darwin's 5 theories, and what was missing, and what Darwin was very painfully conscious of, was any understanding of where these variations came from. These inherited variations. He even accepted the idea that the environment and the behavior of an organism might somehow evoke the origin of adaptive variations, a kind of Lamarckian point of view. And not knowing the origin of these hereditary variations, it wasn't bad enough. It's even worse that the popular idea of inheritance at the time, it envisioned that heredity was based on the mix on fluids. Okay, that's where the notion of having Italian versus Swedish blood comes from, and the notion that whether you're blonde or dark-haired somehow has to do with blood. And where did this come from? By and large, if you were to cross a large dog with a small dog, what you get out is medium-sized babies. Okay, many characteristics in the offspring will tend to be pretty much in between their two parents with respect to some feature of that kind. Okay? And if you think about that, if that was the result of mixing together two fluids, sort of like a dark red fluid and a white fluid, two paints or something like that, you end up getting something in between, like pink, okay? But you're not going to be able to regenerate red or white from pink paint. You can't, okay? It doesn't resegregate out. And so the problem that Darwin faced was, and was pointed out, and one of the criticisms that was made of his theory, was that you would expect all the variation in a characteristic among individuals in a population to be lost over time as a consequence of interbreeding, because extreme types would always be lost by interbreeding with opposite types, giving rise to mediocre, in-between offspring. So everything would end up being sort of a pale shade of gray, if you started out with black and white. So the variation that was the fuel for evolutionary change would be lost. So that's what was missing, okay? That missing element in evolutionary theory was basically filled in, was contributed by the work of Gregor Mendel, who, unknown to Darwin, was at the time doing experiments on peas that resulted in the realization that heredity is based not on fluids mixing together, but on discrete particles. You have no idea what these particles might be, which could resegregate out so that mating two intermediate organisms actually could give rise to two different extremes. Okay. later it was discovered that these particles, whatever they might be, were capable of spontaneous change, and these changes were called mutations. And here we see, for example, the various mutations in the shape and color of the eye of the fruit fly Drosophila melanogaster, and it was found indeed that these mutations arise without any reference to the need of the organism. Usually they're harmful, but every once in a while one might be deleterious. Some of these mutations are quite staggering. Here, for example, I wish the lights were a little bit better, but you can't see it. Here, for example, is a mutation in which the antennae of the fly are converted by a single mutational change in a single gene into legs growing out of the head. So this was the origin of genetics, and it was only after 1900 that Mendel's work was sort of discovered, as it's often put, and that was the beginning of the new science of genetics, which at first was widely thought to be incompatible with Darwin's ideas of natural selection, because the notion was that the mutational process itself could give rise to species, so who needs natural selection? Okay. And it wasn't then until the late 1920s and about 1930 that there was a reconciliation and a union of these two points of view, of Darwin's theory of evolution by natural selection on the one hand being united with the new science of genetics, and it was shown that not only were they compatible, but that the science of genetics now provided the missing link, if you like, the missing elements in a fairly complete theory of evolutionary change. And this was the work, the result of this was, starting in around 1930 and into the 1940s, a period that's called the Evolutionary Synthesis, in which was pioneered first by three individuals, one American, Sewell Wright, and two British mathematicians and mathematical biologists, Fisher and Haldane, the three of them that developed, first of all, mathematical theories showing how genetic changes in populations based on Mendelian principles of genetics, how genetic changes in populations would occur by natural selection and by other forces as well, thereby uniting Darwin's ideas with genetics. The other major contributors, or some of the other major contributors to this synthesis, were Dobzhansky, who was a Russian who immigrated to the United States, who pioneered studying these actual genes in natural populations, wild populations of organisms, especially fruit flies, and actually showing how the genetic variation that was present, how that genetic variation would actually change in natural populations. That, however, had to be bridged to the more classical kinds of observations in biology that had been made by by ornithologists and botanists and zoologists, entomologists and paleontologists. And that bridge was formed partly by Dobzhansky, but also by Ernst Mayr, who was an expert on the diversity of birds and the origin of bird species, Ledyard Stebbins, who played much the same role in botany, and by another systematist, Bernhard Rensch, in Germany, and especially in paleontology by George Gaylord Simpson, who was a preeminent paleontologist of the day. And the conclusion of their interactions with one another and their publication of a number of books was that the new genetical theory of evolutionary change could be synthesized with the observations on diversity of species and their characteristics, as well as observations in the fossil record. And the result of this was a kind of what's sometimes called neo-Darwinian theory, or the new synthetic theory of evolution, which sounds very much like Darwin, except that there's genetics added to it. And so the basic tenets there were that that heredity is based on particles, namely genes that occasionally mutate, that these mutations occur at random with respect to the adaptive value or the need of the organism. Okay? Evolution, just as Darwin said, is a population process by which some of these mutations may increase in frequency within the population, replacing the prevalent form of the gene, or allele as it's called. This replacement can occur actually by two different processes. This was something that was worked out by the mathematical geneticists. One process was natural selection. The other was a process of pure chance, pure fluctuation, purely by chance, but just fluctuations in the relative proportions of the new form of a gene versus the previous form of the gene. Purely random fluctuations called genetic drift. In that case, that would be most likely to happen when the new form of a gene, the new allele, was neither better nor worse than the old form with respect to survival or reproduction. It's said to be neutral, selectively neutral. The so-called the fathers of this new neo-Darwinian synthesis also by that time had established a great deal of evidence for natural selection. And so by that time, in the 1940s, the 1950s, natural selection had been thoroughly documented, and that could be claimed to be the cause of adaptations. Okay. Meyer in particular and Dobzhansky put a lot of emphasis on understanding how new species arise. How does one parental species give rise to two or more descendant species? And that involved an understanding of what species are, and they define species as different populations that don't exchange genes. They're reproductively isolated because of biological differences between them. For example, they might not mate with one another because their courtship behaviors are different, and the females of one might not accept the males of another. Furthermore, just as Darwin said, newly formed species that have come from a common ancestor at first are very similar and only gradually diverge over the course of time. And then they all agreed, and Simpson in particular, the paleontologist, that these principles of slight changes, genetic changes within individual species, those principles indeed could sufficiently explain what was seen in the fossil record. In other words, those could account for long-term, large-scale evolution. So this is kind of a portrait of evolutionary biology a la the early 1950s or even the late 1950s. What happened in the 1950s? Well, a critically important event was, of course, the Watson-Crick model of DNA in 1953. And so this is one of the elements that has contributed to progress throughout biology, of course, and in particular, for tonight's purposes, in evolutionary biology. If you ask, why is it, what has enabled evolutionary biology to advance or to progress? First of all, we have, I keep hitting the forward button instead of the pointer. First of all, as we've already seen, theoretical and conceptual advances, okay? And these have continued. So we've had the elaboration of a variety of mathematical models and other conceptual advances that have really set the stage that essentially constitute new ideas, new understanding, new ways of framing hypotheses that then can be evaluated by experimental and observational work. Secondly, as I will stress, as I've already said, and will stress even more, the development of molecular biology, providing both a deeper understanding of organisms in every way, and very importantly, providing a basis for all sorts of new methods of study. There have been other technical advances, such as computation and more recently informatics, that have also contributed enormously to the growth of evolutionary biology, which is now happening exponentially. And finally, and something I really won't talk about, is the synthesis of evolutionary principles with other fields of study, such as ecology and animal behavior and developmental biology, that have enabled an extension of evolutionary biology into most of the realm of biology at large, and serves to unite the entire field of evolution. So, as I will emphasize throughout, for much of this lecture, the understanding of molecular biology has been absolutely profoundly important to advances in evolution, and I will give illustrations of this as we go on. Okay, so the first, what are some of the I can only touch on a few of the ways in which evolutionary biology has progressed over, say, the last 50 years. Okay, so it's going to be very superficial, but the first point that I want to make is that we now know in a way that Darwin could not know how right he was about the first major point, sort of the first of Darwin's theories, which was the claim, the supposition, his proposition, his outrageous proposition, that all organisms have evolved from a single original form of life, undergoing various modifications in the process of diverging from one another. In other words, the history of evolution, that there has been a history of evolution, of descent from common ancestors, can now be considered a fact. And there are two major sources of evidence on this. The most obvious, of course, would be the fossil record. And you know that continually we're finding new fossils which fill in the gap. You know, Darwin didn't have any examples to really be able to show that there had been transitions from one major form to another through gradual changes. You know, the fossil record was so incomplete, and of course he devoted 2 chapters of The Origin of Species to talking about how incomplete he believed the fossil record was, that he had virtually no examples of intermediates in the fossil record. Okay? Shortly after The Origin of Species, Archaeopteryx, the dinosaur bird intermediate, was found, but that was it. And now we have, just in the last few years, we have this marvelous creature, this Tiktaalik, an addition to our understanding of the transition from fish-like organisms to tetrapods, the first land-living vertebrates, a critically important fossil showing the intermediacy of emergence onto land in the most beautiful way. We now know that indeed birds are dinosaurs, that they are feathered dinosaurs, and we've got this enormous variety of fossils just in the last 15 years or so, mostly from China, showing an enormous variety of forms that illustrate how birds evolved. Here's something that was clearly a side branch in that history. Sorry that it's not a very clear picture, but in the original it is. This is Microraptor, a small dinosaur only about so big that clearly had long feathers used for flight, both on the arms and on the hind legs. It was a 4-winged dinosaur. Okay? So there was clearly an enormous variety of feathered dinosaurs, one lineage of which became, was the ancestor of today's birds. And of course we know that there has been a continual discovery of hominid fossils in Africa. Here, Australopithecus afarensis, showing, if not an exact step-by-step origin of Homo sapiens, at least showing the transition, the alterations in features such as the evolution of bipedality and the shape of the foot, and the reduction of teeth and the size of the cranium, and all of the characteristics that physically define hominids. So there are plenty of intermediates in the fossil record, and don't let any creationist tell you otherwise, which they will do constantly. But even if we had no fossil record whatever, we could be completely confident that Darwin was absolutely right about the single origin of living things, the origin of diversity from a single common ancestor. And that evidence comes largely from comparing living species with one another, all kinds of living species with one another. And the best evidence on that, or the evidence that has made it quite clear what we've known even before there was DNA, it was quite clear that that was the case, but now on the basis of DNA, we can say that with an enormous degree of confidence. Okay. This is a very strange portrayal of a small part of the tree of life. I think you've all seen phylogenetic trees, and usually they have a stem, and a root and branches that come out. In this case, basically the top of the tree has been sort of turned around into a circle in order to fit into a single diagram. So there's a root in here, right in there. That's the root of the tree. And you could unfold this wheel to form an enormous tree with a few thousand branches on it. Okay? This is a portrayal by David Hillis at the University of Texas, who has taken species, from a wide variety of phylogenetic studies of relationships among organisms, and pieced them together into this tree of life, which includes what? It includes bacteria and archaea and all of the one-celled eukaryote organisms, such as things that used to be called protozoans. And it includes all the plants. And it includes all the fungi, which are the closest relatives of the animals, okay? And we may ask, where are mammals in all of this, and where are humans? And the answer is right here. Those little words right there say, you are here. Okay, so let's zoom in on that. And there we are. There is Homo sapiens right next to some other mammals right here. So the DNA sequences confirm that we indeed are mammals. We are closely related to other mammals. And then if you could, if you could see the branches, that they were in the bigger tree, but if you see the branches there, you would see, well, yeah, we're mixed in where we have all the other vertebrates right here as well. Here are alligators and their close relatives, the birds, right here. And then you have lizards and snakes right out here, connected a little bit further down to amphibians, okay, and to these are all amphibians. Hillis works on amphibians, so there are a lot of amphibians in there. He loves amphibians. And back down to lampreys and hagfishes, the most sort of primitive types of vertebrates. And then outside of the chordates are our closest relatives, the echinoderms, the sea urchins right here, and their relatives. And where does all of this come from? It comes from analysis of DNA sequences, okay, that lead us to have real confidence. I mean, we can say today, here is a phylogeny of the relationship between humans and the other apes in which we are really confident. Okay? 20 years ago, we couldn't have said exactly how humans were related to chimpanzees versus gorillas, but today we are really enormously confident of that. What is that confidence based on? Here's just one tiny, tiny bit of information that illustrates this. Okay? These are just little bits of the sequence of a particular DNA sequence which used to be a hemoglobin gene. Okay. But it's a hemoglobin gene that has undergone mutational changes that have silenced it and rendered it basically a fossil, is what it is. It's basically a dead gene, a pseudogene. And here I've shown just little bits of the sequence of it in human, chimpanzee, gorilla, orangutan, rhesus monkey, and spider monkey, a South American monkey, which is more distantly related to any of the others than they are to one another. And illustrated here, throughout this entire thing, most of the sequence is the same in all of these species. Here, for example, here are just a few of the many, 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 many base pairs in that sequence that are identical among all of these species, except for the fact that little set of four elements in the DNA sequence have been deleted, have been lost in the branch that includes Homo and Pan, the chimpanzee and the human. So there is a change in the DNA that has happened that seems to unite human and chimpanzee to the exclusion of all the others. All the others, from a gorilla through the South American monkey, Ateles, they have some DNA there, but that has been deleted there. The same thing down here, down around nucleotide 8,600 or 400 or so, we have another place where you've got a sequence which is much the same for all of those species, but here again, a piece of DNA has somehow been deleted and lost and it's absent both in human and in chimpanzee, and that suggests that it was their common ancestor in which it was lost, okay? And the consequence is that they share this character. There are some other places where you see, where is it? There's somewhere in there I'm looking for, but I don't quite see it. Well, let's not worry about it. Here is one place of many where you see that there's a difference between the orangutan, gorilla, chimpanzee, and human on the one hand, they have an adenine in that position, versus different nucleotides in the more distantly related things. So this would suggest to you that as far as that's one little bit of information which suggests that these four apes, if you like, humans and apes, have had a common ancestor in which there was a substitution, a change in the DNA at that particular place, substituting an adenine for the nucleotide base pairs that you would find in the other species. That's one site in which you see that. Here's another one in which there's a substitution of a guanine. All of those apes share a guanine in that particular place. Here's a place where they all share an adenine again instead of a guanine. And so this is the way in which DNA sequence differences can be found that show which species share alterations of the DNA sequence. And clearly, just from this data, you could say, yeah, it makes sense. The easiest interpretation, and it would be very hard to come up with another interpretation, is that humans and chimpanzees have a very recent common ancestor, that you would have to go further back to find the ancestor of those together with the gorilla and the orangutan, which share some features in common, such as those that I've just described. So, and now another aspect of, so the consequence of that is, we have on the basis of DNA sequences, totally independently of the anatomical data on which zoologists and botanists came to conclusions about relationships among organisms, concluding, for example, that humans and primates are mammals, and that indeed there's a common ancestry between shrews and primates and whales and giraffes. Okay? That was a statement based on morphology, on anatomy, and that is upheld by totally independent data from DNA sequences, including DNA sequences that have no function whatever, such as this dead gene that I've just been talking about. This dead, useless, functionless gene. So we have confirmation of Darwin's fundamental notion about the history of life in a way that he could never have conceived of. Secondly, okay, it turns out that these DNA sequence differences evolve at a very, very, very roughly constant rate. And that means that you can calibrate that. If you have some fossils of certain groups, you can get an idea from the differences between the descendants of those fossil ancestors, you can get an idea of how fast the DNA sequence evolves. And having that, you can then estimate when the divergence happened at certain places in the tree for which you don't have sufficient fossil evidence. And so that it is on that basis, for example, that we have a time scale for DNA sequence differences that enables us to have a rough idea of just when humans diverged from our common ancestors with chimpanzees. And the answer is apparently in the neighborhood of 6 or 7 million years ago. And if you go back to, if you ask, when was our common ancestor with orangutans, the answer is the order of 15 or 16 million years ago. And so we are able to get rough ideas of the dates, the time at which events happened in evolution, which up until the very recent past we had no inkling of, or almost no inkling of. We're able to get estimates of when, for example, different species of malarial protozoans diverged from common ancestors. How long Plasmodium falciparum, for example, the cause of the most serious form of malaria, how long has it been since that species came into existence? Needless to say, there's no fossil record of these tiny, tiny little organisms. So that's a major advance. Now, just as you can infer the historical relationships among species, you can infer the historical relationships among genes, among different versions of a particular kind of gene. In fact, we just sort of did that for hemoglobin. And the consequence of that is we can often say something about the history of populations of a species, such as the human species, based on an analysis of the genetic differences between them. So this is a very famous case of mitochondrial DNA sequences, which show here the relationships among different mitochondrial DNAs taken from humans throughout the world. Oh, I keep doing that. Okay, keep doing that. Okay, there we are. And basically what this tells us, all of these populations which are near the base of this phylogenetic tree, all of those are DNAs from Africa. And that concords, of course, with the fossil record. It says that humans evolved and diversified in Africa. Okay? Which of course conforms to our understanding of the fossil record of humans. And then we have one branch of different versions of this gene that differ from one another by a few mutations. In one branch here, some of those genes are taken from African individuals, and then here are copies of the mitochondrial gene taken from humans throughout the rest of the world. And what does that say? It says that very recently, because the short branches here, the fact that those branches are so short and that they all kind of converge into this one little ladder here, it says that there are very, very few mutational differences among the mitochondrial genes of these different people. And that says there must not have been very much time that has elapsed since those different populations spread out from one another over the world, because that means there hasn't been much time for mutational changes to accumulate between those populations. Okay? So what this tells us is that in the rather recent past, humans emerged from Africa, which is where they originated, as indicated here, spread out very quickly throughout the rest of the world, and very recently. So it says that basically we as a species are extremely recent. There are very minor genetic differences among populations of humans throughout the rest of the world. Okay. Now, this suggests that we spread out from Africa and presumably out across Eurasia, and then we have every reason to believe from Eurasia across the Bering Land Bridge into Alaska and then down through the Americas. And if that is the case, the theory of population genetics that was worked out by some of those guys that I showed in that one picture says that in the process of colonization, if you have just new colonies being formed as they progress across the continent, they're not going to carry all the genes with them. There will just be, if you have just 10 people, they can't possibly carry all the different genes that we in this room, could carry collectively. And so there'll be some loss of genetic variation sequentially, colonization event by colonization event. And if that is true, you predict that the populations that are furthest away from our ancestral source, namely Africa, should have the least amount of genetic differences among individuals in those populations. And very recently, it has been published just a couple of years ago of exactly that is the case. So this is a measure of the degree of genetic variation among individuals in a population as a function of how far that human population is from Ethiopia. Okay? And you see that there's a diminishment of genetic variation exactly as predicted by, number one, the hypothesis that humans originated in Africa and colonized from there, and number two, the theory of the mathematical theory of evolutionary changes, of genetic changes in evolution. Okay, I see that I have a great deal of talk yet to go, which I probably won't get to. Okay. So I've been talking mostly about evolutionary history, and then just at the end here, moving into intimations of evolutionary process. Okay? For example, the process of genetic drift, in this case, the random loss of genes as a consequence of small numbers of colonists progressing across the landscape. Okay? What about evolutionary process? Okay, remember we have the other ideas of how does evolution happen. Okay, we've talked about the history of documenting the history of evolution, and needless to say, a great deal has happened here. One of the most important things that has happened is that we now have tools for analyzing the genetic variation, which you remember in Darwin's theory was the raw material of evolution. That genetic variation somehow arises among individuals in various characteristics. Later, it was recognized that those were called mutations of the genes, and now, of course, we can see those mutations. And so, for example, this is a diagram from a very early study of one particular gene in Drosophila melanogaster, the fruit fly, showing by each of these vertical lines a place in that gene where differences were found, and whether it was an adenine or a thymine or a guanine or a cytosine, where differences were found among genes taken from different individual flies. And of course, it turned out that there's an enormous amount of genetic variation. Okay? Any pair of us in this room have thousands and thousands of differences between us if you go through the entire genome. Okay? No point in belaboring that point. A lot of that variation, as it turns out, seems to have nothing to do with Darwin's theory of adaptation by natural selection. Okay. So remember I mentioned this process that had been worked out by the mathematical geneticists called genetic drift, which is the purely random fluctuation in the proportion of one gene type versus another gene type in a population, this process of genetic drift, by which a version of a gene which is neither better nor worse than the standard version may come to replace the standard version. Okay. And here's some evidence that conforms to that. And at the molecular level, at the DNA level, it turns out that this process of genetic drift has really been important. And the best evidence of is this. If you look at places in the DNA in which any substitution, any alteration of the nucleotide base pair would cause a change in the protein, those are called non-degenerate sites in the gene. And they have shown the slowest rate of evolution. This shows here comparing the homologous gene in different species, for example, human versus rat, okay? It turns out that those are the genes, that those are the places in the genome that show the lowest rate of change, the lowest degree of difference between any two species you look at. And that suggests that an awful lot of the mutations that would happen at that particular place in the gene would cause a change in the protein. The change would be deleterious, it would be harmful, and so it's expunged by natural selection. But it's kind of purifying selection that maintains the same genetic element over the course of the long period of time that has separated us from rats, for example. On the other hand, if you look at a pseudogene, like that dead fossil hemoglobin gene that I talked about earlier, this is an indication of the rate of change. In a pseudogene, it doesn't matter if there's a change in the gene or not. It's totally useless, totally neutral. It doesn't hurt the organism nor harm it. It doesn't improve the organism nor harm it because the gene isn't doing anything. It's a piece of DNA that just isn't doing anything at all. And this is where you see then the greatest rate of accumulation of differences between species that come from a common ancestor. So this was a clear indication that at least at the level of DNA, we have a major evolutionary process going on that is different from the one that Darwin talked about. Okay? He intimated that he thought there could be such random changes, but he never dwelt on it because it certainly did not in any way contribute to his theory of adaptation. So this has been one of the major changes in our understanding of evolutionary processes, which is to say, at least at the level of DNA, that there can be an awful lot of changes that are neither advantageous nor harmful, but nevertheless accumulate within populations and between species. But we also have what Darwin did not have, evidence for natural selection. You realize that in 1859, after he'd been thinking about it for more than 20 years and not daring to write it down, in 1859, Darwin had no actual examples of natural selection in action. Okay? I mean, this was a brilliant idea, which he supported by inference from all kinds of circumstantial observations, but he had no direct evidence. Okay. We today now have hundreds and hundreds and hundreds of studies in which people have done experimental analysis showing that one genotype is more adapted than another, that it survives better or reproduces better depending on environmental circumstances. We also have what Darwin did not have, which is direct documentation of rapid adaptive evolutionary change. Here are just a few examples, okay? One example is this particular species of bug, which has a long beak that it uses for plunging into a fruit to reach the seed inside and then pierce the seed and pump digestive enzymes into it so it can suck out the liquefied seed. In several parts of the range of this particular species of bug, new species of plants have become abundant because they're grown as ornamentals around people's houses, and the bug attacks them, but the fruit size is different. So in some places, it basically has had to evolve a longer beak in order to plunge through a larger fruit to reach the seed within. And in other places, the fruit is smaller, and it's evolved a shorter beak to more effectively reach the seed. Okay, so that's one example. Here's a very famous famous example, the beak of the finch, as Jonathan Weiner entitled his book about the work by Peter and Rosemary Grant in the Galapagos Islands, showing that when there was a drought and a change in the relative abundance of large seeds versus small seeds, that there was greater survival of individuals of this species of finch that had a larger beak. They were able to successfully feed on the large seed that was the only food supply. Here's an instance of evolution by natural selection, but natural selection imposed by humans, which is to say, overfishing. Okay? And what do you do when you fish? Basically, the first fish to get caught, by nets at least, are the larger ones. Okay? Larger means older, usually, in fishes. And so there has been it's like a predator feeding on, and with a huge predation rate, of course, in the case of the Atlantic cod here, the fishery of which has been destroyed by overfishing, but this is like an enormously voracious predator eating the larger fishes, which are those that are most reproductive, okay? And this means that automatically the population is going to consist mostly of the descendants of smaller fishes, namely those that reach maturity and are able to reproduce at a younger age and a smaller size. And so there has been the evolution here. This is the age at maturity, on average, in populations of the Atlantic cod, in which you see the age of maturity decreasing as a consequence of this form of natural selection. And then, of course, we have also imposed natural selection on many, many, many species in a vast variety of other ways, especially by trying to kill them, and especially by poisoning them. And so here we have the number of species of insects and mites which have evolved to be resistant to insecticides since 1940, when DDT was first released as a control agent for houseflies and other pests. And for every class of insecticides, DDT, cyclodienes, all of them, for every one of them, at least 200, on the order of 100 or 200 species of insects have evolved to become resistant. And this, of course, is precisely what we see even closer to home, which is to say the evolution of antibiotic resistance in pathogens, something we're all familiar with, something which is a major, major threat to human health. This is just one of many, many instances in which the proportion of bacteria of this particular species that was resistant to a particular drug increased over the course of about 20 years here, or whatever, 15 years. Just today, I looked in this week's issue of Science and found just glanced at a paper entitled Evolutionary Dynamics of Complex Networks of HIV Drug-Resistant Strains. And it begins by saying HIV resistance to antiretroviral drugs is causing serious clinical and public health problems throughout the United States and Europe. Drug-resistant strains have evolved that differ in their susceptibility to 3 major classes of antiretrovirals, and these strains now are capable of being transmitted to individuals. So you have transmitted drug resistance, which is reported to range between 8 and 22% in many HIV-infected communities. So we have by no means won the war against AIDS, even in the United States. Okay? In fact, this study is applied to the population of San Francisco. So even in the United States, much less South Africa, we are very, very far from winning the war, and the war in part is a matter of the evolution of an organism escaping in this case a virus, or kind of an organism, escaping our attempt to control it. So we have evidence of abundant evidence of natural selection, and now, with the advent of molecular genetics, and especially the advent of genomics, we are often now able to pinpoint the precise gene that is responsible for the characteristic that is evolving, and in some case, the precise place in the gene, the precise nucleotide base. And so, for instance, here are 2 very distantly related species of mice, both of which have certain populations that are pale in color versus dark in color. And in this case, they've been transposed. This one actually lives on a pale environment, and the dark one lives on lava outcrops that are much darker. And the coloration has evolved because basically the coloration is more cryptic and protects them against, to some extent, against predation by owls and other predators. That the point is that this adaptive difference in color in this species of mouse is based on a mutation in a particular gene called melanocortin-1 receptor, and it turns out that that same gene is responsible for a similar difference in coloration in a totally different species of mouse. It is different for the difference in coloration between the white and dark forms of the snow goose, which is a bird that you can see around here quite commonly. That same gene is responsible for the two color forms of this particular Arctic bird, the parasitic Jaeger. And it turns out that melanocortin-1 receptor is also different between humans who are red-haired versus people who have other hair color. So that leads to the very interesting observation that often the same genes are involved in the evolution of similar characteristics in very, very distantly related species. And that has important implications. And that carries through, basically, now that we have basically a readout of the entire genome of many, many different kinds of organisms. That leads us to all sorts of things. For one, interpreting an awful lot of what you see in genomes turns out to really be immensely enhanced by applying an evolutionary point of view. One of the biggest questions we have to ask about genomes is, where do all these different genes come from? You know, I mean, if we've got 30,000 genes in the human genome, and if E. coli bacteria have only about 5,000, where do all those different genes with all those different functions come from? And we can say something about that in an evolutionary perspective. Here, for instance, there is a whole family of genes, the Globin genes, including the globin that's active in our muscles, myoglobin, and the several different hemoglobin genes that we have in our genome. And two of them in particular link together to form the actual functional hemoglobin molecule. There's a beta globin and an alpha globin. And here is a history by which those different genes have arisen by a process of gene duplication. Basically, there's a process by which one gene locus can somehow become duplicated, so there are two versions of it, or two copies of it, more or less side by side on a chromosome, and they then can undergo different mutation and different natural selection so that they can diverge to take on different functions. And that is the case here. Some of these globins are active just in the embryo, and others are active in the adult. They have different functions. And this approach has enlightened our understanding of the evolution of all sorts of different proteins from common ancestors, but proteins that gradually have taken on very different functions, novel functions. So an evolutionary point of view is immensely useful in genomics. And the other reason that it is useful, the evolutionary point of view, is that it turns out that many of the same genes are conserved among really distantly related species and play much the same function. This is one of the most staggering experiments that I've ever read about. Okay. It turns out that there's a gene in fruit flies where it's called the Eyeless Gene, which basically it's like a trigger, it's like a master switch, and it turns on a whole bunch of other genes that are required for making an eye. Okay. You can make extra eyes grow on a fly by doing molecular magic and putting an extra copy of that gene into a fly's genome. And here you have, for example, an extra eye that has grown on the antenna where you're not supposed to have eyes, okay? But it's morphologically a perfect eye. This is a fly, terrible picture of it, but it's a terrible fly picture in which the human equivalent of the same gene, similar DNA sequence, turns out in humans, mutations of that gene cause serious optical defects in the development of the eye. Putting a copy of the human version of that gene into the fruit fly's genome and taking out the fly's own copy of the gene enables that fly to produce eyes, perfectly normal insect eyes, being evoked by a switch gene, which in this case is the human switch gene. So what that says is, here is this gene which is playing the same function in insects and invertebrates, whose last common ancestor is at least 500, 600 million years ago, probably closer to a billion years ago, since we've had a common ancestor with insects. And it is still playing the same function, and the subsidiary genes, the downstream genes, can still read the message that's coming from a human gene and say, oh yes, I know what to do, I know that message. It's the message that the insect gene normally sends. So this is an extraordinary thing, and it turns out that there's this sort of so-called genetic toolkit of this kind, so that many, many aspects of development have a genetic foundation, which is, in large part, the same among really distantly related organisms. And this means that you can learn an awful lot about function of detecting the function, the hitherto unknown function of genes in the human genome by studying genes of flies, okay? Or genes of C. elegans nematode worms, or some genes of yeast even. And that, of course, is the justification for studying model organisms in which your research that is funded by the National Institutes of Health is not really directed at understanding the fruit fly or the C. elegans worm or yeast. They really don't care about yeast. They really care about understanding human function, and the supposition is that we can learn about human function by studying the fundamental molecular biology and cell biology of a vast variety of very distantly related organisms. And the only rational scientific justification for the assumption that we can learn about humans by studying yeast or worms or flies or even mice is that we have common ancestors, that we share the same fundamental processes as a consequence of our common ancestry, of our shared ancestry with these other organisms. So that we have all inherited from our common ancestors the same fundamental properties. Okay. I think that I'd better skip over this wonderful section on the science of adaptation, in which, for example, Darwin's notion of sexual selection, a fascinating idea, has been thoroughly documented and justified and leads to all sorts of interesting studies. Understanding how cooperation can evolve in what is supposedly the world of nature red in tooth and claw. Well, that's something that we'll have to wait for another day. And just end by saying that, of course, we've made immense progress in so many different ways, but it's certainly not as if we now know everything that we need to know about evolution. There are many areas in which our ignorance is absolutely staggering. And I could give a long list, especially if I knew more about molecular biology and genomes. I could give you a very long list of things that we have yet to learn about the evolution at the genomic level. But at the more phenotypic and ecological level, the organismal level that's more congenial to me, there are things we don't know. How really do species evolve in response to one another? Predator and prey, parasite and host. Do we really understand which, how they're able to maintain coexistence if each of them is evolving to foil the other? How does adaptation to really new environments happen? Really, I mean, whales. How does a land-living mammal first take up an aquatic existence, and then such that it puts itself in a position to be subject to natural selection for greater and greater adaptation to an aquatic environment? We really don't know whether adaptation is limited by the supply of advantageous mutations. And then finally, it turns out that a big problem in evolution is not that, how could all that evolution possibly happen in so little time? Because actually there's been lots of time. The big problem is understanding why evolution is so slow. And I will finish with that. That is to say, we have phenomena that are very hard to understand, given that I've already shown you that adaptation can often be very, very rapid. We nevertheless then have very paradoxical observations. Okay? For example, there are about 80 species of butterflies in the particular group called the heliconiine butterflies. They've descended from a common ancestor that's on the order of 40 to 50 million years ago. And all of them, when they are caterpillars, feed on plants in one family, the passion vines. Why haven't they evolved to use a greater variety of plants? There must be lots of instances in which populations of butterflies have faced, sort of a paucity, of passion vines. It's not like you can find passion vines anywhere you go. So why haven't they become more capable of using a greater variety of potential foods? Okay. Eldridge and Gould, in their famous paper on punctuated equilibrium, called attention to what they called stasis the fact that most sequences in the fossil record show actually very, very slow evolution, or virtually none at all. Here's an extreme case, a particular species of clam, in which this is the living organism. Here are fossils taken all the way back to 17 million years, and to all intents and purposes, they're identical. Why is that? Can it really be that they've lived in the same environment the environment hasn't changed for 17 million years? Okay? And I would have to say that we really don't understand that. And then, of course, finally, we have the problem of extinction. If it is the case, as we seem to have, we have a great deal of evidence for the capacity of species to evolve, to adapt very rapidly. If that's the case, why are there so many losers? Okay? Far more than 99% of the species that have ever lived, of course, are extinct. Okay? And that, in the current day and age, is a problem that really should concern us. Because, of course, we are right now in a period in the history of the Earth in which the environment is changing just about as fast as it has ever changed before in history. And that, of course, is because of all the changes that we humans are wreaking on it. In so many different ways. I mean, there's pollution, there's overhunting, there's fishing, there's acid rain, and of course there is global climate change, which, despite the skeptics, is very, very real. And so I expect that this beautiful ivory gull that lives up in the icebound north is going to be extinct within a century or so. And so will these wonderful plants at the tops of tropical mountains, because there's no place to go once it gets too warm for them up there, unless they adapt to the greater temperatures. But it's not at all clear that they will. And then we have species that have been diminished by loss of their habitat, such as prairie chickens. We have species that are being extinguished by diseases that were spreading around the world, such as these tropical frogs, which have been devastated by a particular kind of fungus that threatens to wipe out hundreds of species of frogs in the tropics. And so we are, humans are creating changes in the world that are very possibly going to result in one of the largest extinction events of all time. And it would be nice if we had some understanding of when species are capable of evolving rapidly, which we often see happen, and when they are not, because clearly very often they're not. And if there's one large, looming, impossibly difficult question that remains unsolved in evolutionary biology, that is it. So we have made immense progress, but there's a great deal of work yet to be done. And the study of evolutionary biology, which, as you can see, unites really all the biological sciences. It is relevant to everything from understanding DNA and genomes all the way through ecological problems such as the extinction of species. The study of evolutionary biology is both unites the biological sciences, has immense implications for humans, for ourselves, and clearly has a long way yet to go in accomplishing its aims. Thank you for your patience.